everyone, my name is NAJWA binti KHIRUL ANUAR, and today I will be presenting on the topic gut microbiome and human health. This presentation will explore the fundamental of the gut microbiome, its connection to COVID-19 infection, and the technology used to study it. The gut microbiome is a complex and dynamic community of microorganisms, including bacteria, fungi, and viruses that live in our gastrointestinal tract. This community considered as a key factor in regulating our overall health. From birth, this community evolved and continued to influence our well-being throughout life. The diversity of our gut microbiome develops until early adulthood and then becomes relatively stable. Interestingly, its diversity peaks around age 65 and then begins to decrease, with a more noticeable decline in people over 80 years old. Development of our gut microbiome may begin before birth, with early exposure from the mother. After birth, a baby's microbiome is shaped by several factors, including the mode of delivery, feeding method, and early environmental exposures. By adulthood, the gut microbiome becomes remarkably personalized and stable. It doesn't drastically change with short-term dietary shifts and characterized by a high number and abundance of different species. As we age, the diversity of the gut microbiome often decreases, especially in older adults, potentially contributing to the age-related health issues. Many factors influence this community, including diet, physical activity, antibiotic, pollution, stomach acidity, bile acid production, obesity, and the immune system. A healthy gut microbiome performs several vital functions for our bodies. It aids in digesting complex carbohydrates, maintains the integrity of the gut barrier, synthesizes essential vitamins, and regulates the immune system. It prevents colonization and invasion by pathogenic bacteria. An imbalance in the gut microbiome is known as dysbiosis. A 2024 study by Sun et al. Revealed that the composition of the gut microbiome is significantly disrupted in many diseases, including COVID-19. The virus SARS-CoV-2, which causes COVID-19, primarily affects the respiratory system. However, recent findings show it profoundly impacts the gut microbiota. This disruption can contribute to disease severity, systemic inflammation, immune system imbalance, and increased risk of chronic diseases. How microbiome imbalance affects COVID-19? Let's look into its mechanism. Firstly, the virus infects gut enterocytes by binding its spike protein to the ACE2 receptor. Transmembrane protein serine to then facilitate viral entry and membrane fusion by allowing the virus to replicate inside the cell and introduce new virions that infect neighboring cells. This infection causes cell death and triggers strong inflammatory response and disrupts tight junction between cells, compromising the gut barrier. This results in gut dysbiosis and increased permeability, often known as leaky gut. Leaky gut allowing pathogen and toxin to enter bloodstream, which further causes serious complication. Second mechanism is through ACE2 depletion and disruption of the renin-angiotensin-aldosterone system. Angiotensin-converting enzyme 2 is crucial in absorbing essential amino acids, including tryptophan, and play a role in counterbalancing the effects of angiotensin 2 in regulating blood pressure and inflammation. Tryptophan is needed for serotonin synthesis, antimicrobial peptide production, and maintaining a balanced composition of gut microbiota. Binding of the SARS-CoV-2 virus to the ACE2 receptors lead to the internalization and downregulation. This impairs the absorption of the tryptophan and causes dysbiosis, which allows for the overgrowth of the pathogenic bacteria. The downregulation also diminishes the conversion of angiotensin 2 and promotes increased intestinal permeability. This increased permeability allows for the translocation of the bacterial endotoxin from the gut into the systemic circulation. This process fuels systemic inflammation and a key factor contributing to the severity of the COVID-19. Next, the consequences of dysbiosis in COVID-19 are significant. First, dysbiosis causes microbial translocation, which further contributes to intestinal inflammation and barrier dysfunction. This biosis also causes immune dysregulation and disrupts the balance of immune cell population and triggers local inflammations, weakens the intestinal barrier, and creates a cycle of ongoing intestinal damage. Second, dysbiosis induced immune dysregulation via the gut-lung axis significantly impacts the pulmonary microbiome ability to maintain hemostasis, leading to respiratory injury and post-acute sequelae of COVID-19. Microbiome translocation triggers excessive pro-inflammatory cytokine, which further enhance lung inflammation and increase susceptibility to acute respiratory distress syndrome. Third, this biosis disrupts the gut-brain axis where it impairs production of short chain of fatty acid, including butyrate, exacerbates inflammation, and compromises the integrity of the gut-brain communication network. This biosis also alters neurotransmitter synthesis, including serotonin, GABA, and dopamine, affecting mood regulation and cognitive function, leading to anxiety, depression, and neurodegenerative diseases. One method to manipulate the gut microbiome is known as clustered regularly interspaced short palindromic repeats Cas system, or known as CRISPR-Cas. The principle of this method, CRISPR-Cas system, are coded in special loci in the bacterial and archaeal genome known as CRISPR array. These arrays are made up of series of short repeated sequences separated by unique spacer sequence. The spaces often demonstrate similarity to genetic mobile elements such as pages or plasmids. This feature reflects the space's origin and provides the system with the main function to defend against invading nucleic acid. When a bacterium survives an attack, the unit of a new spacer and an additional copy of the direct repeat is inserted between the latter sequence and the array. This chronological record allows the cell to quickly recognize the defend against subsequent attack from the same type of invader. CRISPR-Cas system offers a promising approach to targeted gut microbiome manipulation. This system has high efficiency, flexible, and precisely targeted gene editing methods. Is able to manipulate and differentiate genetically heterogeneous bacteria and allow an elimination of specific target bacteria by either engineering lytic bacteriophages or delivering CRISPR-Cas system directly. Probiotics are being engineered with CRISPR-Cas system to enhance functionalities that are more beneficial metabolically. This is an example application of CRISPR-Cas system. In this study, a CRISPR-Cas-based genome editing tool has been developed to manipulate bacteroid species, the most abundant bacterial genome in the human gut. This technology, utilizing the CRISPR-Fn-Cas12a system, enables both markerless gene deletion and insertion. In the key applications, researchers delete large metabolic gene clusters in bacteroids Tetraiotomicron to facilitate functional studies and development of engineered prebiotics. This precise manipulation is crucial for advancing mechanistic understanding of the gut microbiome and for the future development of engineered life biotherapeutic such as prebiotic. Next, this study utilized CRISPR-Cas9 as an antimicrobial as a promising strategy to precisely eliminate targeted bacterial population with minimal impact on the rest of the microbiota. In this study, an engineered conjugative prebiotic strain was used to deliver a CRISPR-Cas system via the conjugative plasmid TP114, resulting in the complete clearance of antibiotic-resistant bacteria from a targeted population in situ in mice. As a final note, the information presented today was drawn from the following key resources. Thank you for your attention.